Yes, I am from Texas, although I was born and raised in England, and therefore I won't be able to convey quite the energetic enthusiasm that was incorporated into Ted's talk, much too restrained for that. But this is starting with so all of my the contributors to this effort that I'm going to report now are listed here, and it's really been a tremendous team effort to work together on improving the Brown Norway reference assembly. And I like this particular West Texas picture. This weather phenomenon is called a haboob. Many people just call it a dust storm. But I like the fact that it's brown and it represents this kind of foggy area between the solidity of the ground and the heavens above that we would like to try and clarify to complete as a current and technologically perfect Brown Norway assembly as we're able to. So Kai has already mentioned some of these pathways. I'll use this slide just to say that the data that we have used to make this assembly has drawn heavily upon other people's work. The short read sequencing came from the Medical College of Wisconsin. The PacBio CLR, the ARIMA HiFi, and the BioNano data came from Sanger. We built a hybrid map with the BioNano data from our own configs with help from people at Texas A&M University, and Melissa Smith has been mentioned, did all the HiFi sequencing for us. So none of this would be possible. The bioinformatics pipeline, you heard a talk from Kai. That was his first public talk, I think, in English. And I think he did a great job describing and explaining this pathway. And it's so wonderful that we can bring a new generation of scientists who come almost ready-made with all of the computational skills necessary to jump on board and do this. Whereas I'm still up here scratching my head, could I ever do such a thing myself? So Ted just mentioned some of these scores. Something probably I have to pay attention to here. So these BUSCOs represent about 10,000 genes, and they're universal orthologs, highly conserved. They're single copies, but they don't represent the whole genome. So you can get quite high numbers using the regular BUSCO software that does alignment with a different one than the one that Ted just showed you. They're not as good as those. This is when you use a database of the Gleyer's conserved orthologs, you get similarly high or better numbers. And I just wanted to show at the bottom here, if I can find the mouse that will actually move, I can't move my mouse so well, is the T2T CHM13, which is the hydatidiform mole. And that's a human haploid cell line. And that was done T2T, and you can see that its completeness and fragmented scores aren't even at the beginning of the fact. So we're doing pretty well. Compiasm Ted mentioned, it's the newest version that uses miniprot as an alignment tool. It's more, first of all, it's a lot faster, done in hours rather than days, and also it's probably better at capturing alternative splice sites with the BUSCO gene assemblies. And so those metrics, Ted's shown you some of those. They're very good for some of the older people around here. If you just look at the list of the top panel, surprisingly, we've come a long way since RN4. RN6, measured by BUSCO, was very, very good indeed. RN Solera, which is a very old assembly, is remarkably good. And I think many of us sort of discounted that assembly because it wasn't entirely, it was public money, but a non-academic source. So BUSCO's take you so far, but we've been fortunate that Terence Murphy and his group at NCBI have used their automated annotation pipeline, which uses the NM and NR-RefSeq genes for the rat, and applied this methodology both to what will become the next rat reference assembly, which we call GRC, since it's been accepted by the GRC consortium, and we'll call it R8 since we had 7 before. Applied it to both. Similar number of transcripts and genes were therefore applied to the assemblies. And clearly we're doing pretty well across all of these different categories. Unaligned transcripts would be problems. We have 12 versus 18. Split transcripts, none in either. Low coding sequences coverage, quite an improvement there. Drop transcripts about the same. So as you probably know, the biggest problem with the MRET BN 7.2 that they've been doing curation work on is with frame-shifted coding sequences that really represent a problem arising from the CLR reads. And use of HiFi has gotten us down to the relatively low number of 18 coding sequence gene frame shifts affecting 14 genes. We're at the point now where the problem is just as likely to be with the ref-seqs themselves as are with our assembly. And so forth. So total number of problems, we're down to 104 versus the 300 that we were at before. And Ted has now reminded us that NCBI would like you to wipe the dirty feet of your genome assembly on the doormat before you enter their archives, and we've been doing that. Ted showed you this data. He went over it quickly. I'll show you something a little bit more that will illustrate it better. He didn't show you brown Norway, so it's good for you to see that. So I think these are Arabidopsis, and it's an assembly from these strains that have been crossed. And so these chambers here represent two different haplotypes, and then there's a separate haplotype here. These are reads that are found only in the assembly, and so they're an error. And these are reads that were in the read set, but are not found in the assembly. When we come over to brown Norway, we don't have any reads that are found only in the assembly. And the number of reads that are found, the chambers that are found only in the reads and not in the assembly is extremely small.
Are those read only? Are those basically centromeres?
I can't tell you. I don't know. I'll show you a little bit more about centromeres in a minute. With the product in 7.2, we have to, okay, and so you have an alternative assembly as well as the is a fully inbred strain. This demonstrates that clearly. And this is completeness, 100% coverage, and QV values that we've talked about that are extraordinarily high. So I'd just like to spend some time now talking about differences between the assemblies. So we have added to every chromosome, but you'll see here that some are highlighted in red, where we've added quite a lot to those chromosomes. And as I go through, I'll explain to you why that is. You've seen this before. So 7.2 is a very good assembly, and we were very grateful to have it. But there are some important differences that are emerging, and I want to walk you through some of those. But before we get into those bigger structural differences, just let's look at some short sequence differences. And so the major differences between the two assemblies are in indels and quite a few SNPs. You will recall that part of the discussion about BN 7.2 was that there was a very large number of variants. And each of these variants were shared by a large number of strains, but they were different in BN 7.2. And that suggested that they represented errors in BN 7.2, and that's why all the other strains were different. This is what we have now. It's a remarkably small number. In fact, I almost think it's too small. It ought to be bigger, just to reflect some drift in the BN genome. But that's where we are. So one of the things we want to do is to try and evaluate whether the potential problems in the assembly, particularly with respect to coding sequences, represent real problems or whether there's an error that's creating them. And so Hao has shared some data along with what I just showed you, looking at the distribution of course, we have a haploid assembly, so these heterozygotes are in their long-range reads, as well as some homozygous differences. And what we've been trying to do is identify those homozygous differences that are in several other brown Norway reads and look at those that fall within coding sequences in our assembly. Of course, most of them don't, but some of them do. And so here you see 4 panels. The variant call file that we produced from those 4 BN Illumina read sets is demonstrated here with a little marking where the variant is. And down below that are the isoseq reads that we've taken from brown Norway, along with the assembly. And first thing to notice here is that, of course, the isoseq reads and the brown Norway, the genome assembly, are 100% concordant here. These are collapsed reads. There's a total of about 70 reads here, although you only see 2 lines. But they're also falling at one of these homopolymer repeats, which is where HiFi sequencing still has some problems. Indeed, I think all of these that I show you here are homopolymers. 3 out of the 4 are clearly, we've gotten it right, because the isoseq reads in the brown Norway assembly are concordant. Some of them remain ambiguous because isoseq has a problem with homopolymers, just as does the assembly. And among these other 6, we've got it right out of 5, and 1 is ambiguous and probably wrong. But we're doing pretty well with those, I think. So let's go back from the single base level kinds of problems to look at what we've achieved at the chromosome scale. And there really are 4 elements that diverge between the 2 assemblies, the first of which is centromeres, Rob. And so I'm showing you the ideograms here, as well as these alignments, syntonic alignments between brown Norway and the upper brownish line and the dark gray line below. And the solid gray regions between those lines are syntonous regions. And when you see white spaces, that space is where we've added sequence in the new assembly. Chromosome 1 is submetacentric with the centromere at about 40 megabases, and we've filled in a bit of the centromere there, but there's a couple of other blocks just upstream from that. Chromosome 10, we haven't added very much to. Chromosome 11 and 12, you'll see in both cases, at the telomere, there's a large amount of added sequence, and I'll tell you what that's all about at the moment. Chromosome 14, we seem to have added at the centromere as well. I'm not sure that they're complete. I'm not even sure if they're accurately assembled, because those are the centromere satellite sequences, and they're highly repetitive, but they are at least assembled and inserted in there. So you see that the centromeres are being filled out here, and you also see that the sequences that we're adding to the assembly are being added in contiguous regions. They're not sprinkled all over the place. Chromosome 19 here, very interesting. We filled in a little bit of the centromere, but that's the small white area. The big white area I want to tell you about. We can go through this. Chromosome 3 is like 11 and 12, and I'll explain that one to you in a minute. And then chromosome 9 is another example where we filled in something that wasn't there before. Chromosome Y, we have gone from an 18 megabase chromosome Y to a 60 megabase chromosome Y. I think we're quite close to completion. There are some unassembled components that derive from Y that may make it bigger eventually. So let's talk about rDNA arrays. These were the main motivation of one of the main motivations of the Human T2T project. These are arrays that contain a lot of repetitive sequence, and in the rat, they occur on chromosomes 3, 11, and 12. And these are acrosomal chromosomes, and the rDNA arrays are in that terminal acrosomal region of the chromosome. And in fact, because these are responsible for Robertsonian recombinations, this is sort of an inherited trait by which different inbred strains differ in how much of the ribosomal arrays are on different chromosomes. So I've outlined brown Norway there. Just to the left, you see that F344 has most of the signal for these arrays. On chromosome 3, brown Norway is more on 11. So here's chromosome 3. The place that we added to in chromosome 3 is where the rDNA arrays are. Chromosome 11, the same. Chromosome 12, the same. So we're getting into some of these difficult and complex repetitive sequences. Here's chromosome Y. Here, I can't really compare in a useful way brown Norway 7.2 to our HiFi assembly, but let's look at it compared to the long read based SHRSP assembly. And you can see in the proximal half of the Y chromosome, there's great syntony between the 2 assemblies. And then you get down into the heterochromatic region where it becomes much more jumbled. But even in that region, there are areas that are quite similar. In the last few weeks, there's been an assembly of 43 human Y chromosomes published, and they really show a kind of similar, this is T2T based work, and they show a kind of similar pattern where in the proximal part of chromosome Y, there's a lot of similarity between each of these Y chromosomes, but as none is the heterochromatic right-hand side, they don't even demonstrate that because it's such a jumble. But they do point out, and maybe we'll find this as we get better Y chromosome assemblies in the rat, that human Y chromosomes vary substantially in size. So it's almost a range of 2-fold. This is really a fascinating biological story that comes from our analysis that will explain some of those other gaps that we filled in. Sex chromosome competition. This was a phenomenon well documented in the collaborative cross lines, where certain collaborative cross lines have a meiotic drive that leads to the overproduction, if you like, of male or female progeny. And those colored dots of blue and red that you see here represent lines that have this distortion. And this distortion is not geographically determined. It occurs at JAX, it occurs at UNC, and it doesn't appear to be mediated by maternal effects. It's not not driven by sex-based neonatal mortality, and neither does it seem to be Mendelian. It seems to be a multi-locus phenomenon. So as we look at some of these areas that we've filled in and ask ourselves what might be in them, here on chromosome 19, we have this 17 megabase segment that we've added to what was a small chromosome to begin with. And there are these genes called spear genes, which are sperm expressed genes. They're expressed only in the testis. And there were a few of them that were incorporated into BN 7.2, but we've replaced them, and this area has been completely expanded. In comparing BN HiFi to SHRSP, BN HiFi seems to be much bigger in this region, but SHRSP has been expanded here as well. So what are spear genes? It's a rodent-specific mouse and rat highly expanded gene family. In the mouse, the comparable region has 215 copies of these spear genes, and it's testis-specific expression. And they've been implicated in this phenomenon of sex chromosome competition that is the basis of the meiotic drive for sex distortion in progeny. Their expression is controlled by Y chromosome transcription factors. I couldn't find any rat spear genes in GenBank. We took isoseq transcripts from testis. We don't have the BN data yet, but we did use the SHR testis transcripts. And they align across this chromosome 19 region. You can see about 4 different exon patterns across these genes. And so I took 4 of them as sort of model transcripts and blasted them to our assembly and found 771 high quality hits. So there has been vast expansion of these genes as a result of this process of sex chromosome competition. Some of these other regions that we've expanded are also full of these genes. So they typically begin with SPE, there's a sperm and testis expressed gene family in this case. They're located there. There's a sperm motility kinase family of genes. Here are some of the transcripts. I think we have 11 transcripts, testis expressed, and you can see that there's a little bit of SNP variation across them, but it's a highly closely related family of genes in this area. And here's another, this is prostate and testis expressed gene family. I want just a few more minutes, if I may have them, to show. So we've been going through this conference and talking about mapping, particularly of mice. And we've seen what high density SNP maps we have. But we don't really know what variation occurs structurally at the local small scale underneath some of those SNPs. We say, oh, this gene is there, that gene is there, but sometimes there's more than meets the eye that we haven't been able to see yet. So this is a tool for looking at non-allelic homologous recombination. It's pretty good for looking up to about 10, 12 megabases. So I've done 2 panels here that are sequential across the proximal region of chromosome 17. So we're covering about 32 megabases. So if you look at the upper right-hand corner, you see the XY coordinates between Brown Norway and SHRSP, and they make a very nice midline. Well, you see these off midline areas, these represent some sequence similarity and represent some evolutionary history with recombination in this region. But as we move down that line toward the telomere, I pointed out this very dense box on the XY, and you can see there that this diagonal line is broken across this region. And it looks like a bunch of parallel lines, and those represent duplicated copies, and it just happens to be a locus where there's more of these testis expressed sperm genes. But we can apply the same approach to other areas where we know there's structural variation. For example, the MHC class 2 locus on chromosome 20. SHRSP and SHRB2 are believed to belong to the same haplotype group in this region, and we get a nice, although there's background stuff in there, we get a nice XY coordinate in this region. But Brown Norway is different from SHRSP, as is WKY, and you can see the structural recombination events that have taken place there. So that can be important for us if we're focused on these areas, because things are not going to look the same in a strain that we're comparing to a reference in these sorts of regions. Look at this region, an area that I'm particularly fond of, which is an adaptive immune receptor locus. In fact, in the rat reference, the immunoglobulin adaptive immune receptor genes and the T cell receptor genes aren't even annotated. And one of the reasons why is probably because of the great diversity that exists here. So in this region, you'll see that on the x-axis, brown Norway is covering about 4 million bases, whereas SHRSP is covering 2 million bases. And the reason why is there's a lot more sequence in there. And the reason why there's a lot more sequence is a lot of sequence duplication that's taking place in brown Norway. So when you have structurally accurate whole genome assemblies, you can dig down underneath potential mapping peaks and explore whether structural variation has occurred and whether it may be relevant to the phenotypes. This one drives me bonkers. This is the constant region of the immunoglobulin heavy chain, SHRB2 to WKY to brown Norway to WKY, all look pretty good. But in SHRSP to everything else, there's this huge block of structural variation, which I could tell you a lot more about. It's really a mouth-watering biological tool. And I'll just end with this picture because I think this is so pretty. This is again a region we filled in that has these sperm motility kinase genes in, where you can see not only duplication, where SHRSP has more sequence here than brown Norway, but you can also see the inversion, the combination of events that have led to past inversions. So you have these sort of crisscross patterns, which I think is just wonderful to both provide you biological insight into the model you're using and to see evolution at work under those loci. And just to end by thanking the people who made so many different types of contributions to this work. Thank you.
Great.